Hello and welcome to this MRC PCH revision video. Today we're going to discuss the kidney's role in acid-base balance. So here we have a picture of the nephron, and the kidney's role in acid-base balance starts in the proximal convoluted tubule. So let's take a closer look. So this diagram shows the lumen of the tubule at the top, the cells of the tubule wall in the middle, and the paratubular capillary at the bottom. So let's go through the sequence of events. So at the glomerulus, protons and bicarbonate ions are freely filtered like this. And in the tubular lumen, they combine to form carbonic acid. On the luminal side of the tubular wall, the tubular cells have on their surface carbonic anhydrase 4. And when carbonic acid comes into contact with this, it's split up into water and carbon dioxide. That then joins up with carbonic anhydrase 2 inside the cell and dissociates back into protons and bicarbonate. So what happens next is chloride from the circulation joins up with the bicarbonate at a transporter and is exchanged. So chloride goes into the cell and bicarbonate is reabsorbed into the blood. So what about the protons that are generated? So the first one, there is a proton pump, so ATP, an ATPase that actively secretes hydrogen ions into the tubular lumen. Sodium is also freely filtered, and there is a sodium proton exchange on the luminal surface, and the remaining protons are exchanged for sodium. The bicarbonate is then absorbed back into the circulation, and the protons are excreted in the urine. So we'll recap that again on this side. So back again, the filtered proton and bicarbonate joins up to form carbonic acid, that then joins up with luminal carbonic anhydrase 4 to form water and carbon dioxide, which is then taken up by carbonic anhydrase 2 and converted into protons and bicarbonate. The bicarbonate is then exchanged with chloride from the circulation, and the bicarbonate is reabsorbed into the circulation. And the protons are either actively secreted by a proton ATPase or exchanged for sodium from the filtrate. The protons are then excreted in the urine, and the bicarbonate is reabsorbed into the blood. So the proximal convoluted tubule accounts for roughly 70 to 80% of the reabsorption of bicarbonate back into the circulation. So we'll now turn our attention to the distal convoluted tubule and the collecting duct system. So here we have two types of cell. These are called intercalated cells, and there is a type A or alpha cell and a type B or beta cell. So the type A cell is responsible for secreting protons into the urine to make the urine acidic. And it also accounts for the remaining 20 to 30% of bicarbonate reabsorption. So it's very similar to the proximal convoluted tubule cells in that the carbonic acid formed from the filtered protons and bicarbonate is taken up by carbonic anhydrase 4, converted into water and carbon dioxide, which is then converted by carbonic anhydrase 2 in the cytoplasm to protons and bicarbonate. And just like on the proximal convoluted tubule cells, we have the proton ATPase, the sodium proton exchanger, and the bicarbonate chloride exchanger. Unique to the type A intercalated cell is a potassium proton exchange ATPase. So here you can see that in the type A cell, we can secrete the protons generated by the carbonic anhydrase into the urine in exchange for sodium, potassium, and it can be actively transported across on its own. And just like in the proximal convoluted tubule, the bicarbonate is exchanged for chloride and reabsorbed. So type A intercalated cells are active when we're in a state of acidemia in the blood and want to be rid of excess protons and retain bicarbonate. So as you can imagine, the type B cell does the precise opposite. So again, through the carbonic anhydrase system, we end up with our proton and bicarbonate absorbed back into the cell. And here, just like on the proximal convoluted tubule, but in a mirror image, we have the same transporters. And again, but in reverse, we have the potassium proton ATPase. So it does exactly the same job as the type A cell, but in reverse. So instead of reabsorbing bicarbonate and secreting protons, here we secrete bicarbonate and reabsorb the protons. So the type B cells act when we're in a state of alkalemia in an effort to correct that. And that's, in a nutshell, how the kidneys regulate our acid-base balance. This becomes important when we start to consider renal tubular pathologies, and we'll consider those in future videos. Thank you.